Hello students, myself Dr. Akhilesh Kumar, Associate Professor of Botany, BCGCW Nagal Chaudhary. Today I am going to speak on lysosomes. This is the one of the very important cell organelles. Uh, just I want to know that what is the lysosomes? Lysosomes are spherical organelles that contain enzymes like acid hydrolases. They breaks up food so it is easier to digest. They are found in animal cells while in yeast and plants the same roles are performed by lytic vacuoles. Lysosomes were discovered by the Belgian cytologist Christian D. Douay in 1955 and named as suicidal bags or suicide's bag. Origin, how it is originated actually. Lysosome may originated from the endoplasmic reticulum and Golgi membranes from the process of budding off. Novikov in 1960 found that acid phosphatase, one of the characteristic enzymes of the lysosomes. Just how where it is present actually. It is present in most of the eukaryotes, but it is absent in prokaryote. Do not have lysosomes, but some hydrolases have been located outside the cell. Lysosome found in higher animals. Lysosomes actually are globular and range in diameter from zero point 0.2 to zero point 0.8 microns. Under the electron microscope, lysosomes appear dense and finely granular. Lysosome is essentially a sphere of lipoprotein membrane containing enzymes. The membrane insulate the enzyme from the rest of the cell. If the enzymes are released, they can digest the cell and hence lysosomes are sometimes called suicide gland. Lysosomal membrane actually. Lysosomes are bounded by a single lipoprotein membrane. The membrane is impermeable to substrates of the enzymes contained in the lysosomes. Certain substances called labilizers caused instability of the lysosomal membrane, leading to release of enzymes from the lysosomes. Lysosomes now comes on the structure of the lysosomes. Other substances called stabilizers have a stabilizing action on the membrane. Labilizers examples vitamin A, vitamin B, vitamin K, vitamin E. And another example of labilizers is cholesterol and cortisol. Third point is that molecular weight and molecular structure play an important part in the permeability of the lysosomal membrane. Compound having a molecular weight higher than 200 do not diffuse through the membrane. Just we can see the very clear cut structure of the how the lysosomes uh, formed. This is the Golgi apparatus and it circulates just like a circular structure that is called the primary lysosomes which fuses and form the secondary lysosomes. Nextly, the lysosomes contain these type of enzymes. Number one is the nucleases, which includes ribonuclease, deoxyribonuclease, breakdown polynucleotides into nitrogenous bases, phosphatate, and pentose sugar. Another important enzyme is that phosphatase, acid phosphatase break down phosphate esters to monophosphates. Lipases that break down then into the fragments, lipids actually, and proteases, protein breakdowns into their constituent amino acids, and glycosidases that beta glycosides, beta glucuronidase and monosaccharides break down into the polysaccharides to monosaccharides, and some other important sulfatases act on the sulfur esters breaking down into the fragments. Just how it polymerizes and how it comes in the different forms. The lysosomes shows polymorphisms in different cell types and even within the single cell. Previously, there are two types of lysosomes. Number one is the primary and secondary lysosomes. The later on of three times, it means changed the structure like phagosomes, residual bodies, and autophagic vacuoles. And next, just you can see that the primary lysosomes is formed from the endoplasmic reticulum and Golgi complex. The enzymes of the primary lysosomes are synthesized by the ribosomes. It can secrete its enzyme outside the cell or store them. Next point is that phagosomes is formed by phagocytosis and pinocytosis of foreign materials. The phagosomes associate with the primary lysosomes and undergoes fusion to form the secondary lysosomes. Next, polymorphized body is that residual body in the final particles containing indigestible materials. It is formed when digestion is incomplete. And fourth and last one is that autophagic vacuoles or autophagosomes is lysosomes containing some parts of the cells in the process of digestion. Just we can see that how the different phases, how the primary lysosomes converted into the secondary lysosomes, and just you can see in this structure, just during the phagocytosis the residual bodies comes into the cells through the plasma membrane and fuses with the primary lysosomes and converted into the secondary lysosomes, and it goes outside. Lysosomes is essentially a bag containing digestive enzymes. The enzymes may be released within cell itself or outside the cells. Lysosomes serve several functions, important functions. Number one is that uh, extracellular digestion. Number two is the intracellular digestion. Third is the hormone secretion, fertilization, autolysis, and chromosomal breaks. Just you can see that lysosomes just extracellular digestion of extracellular matrix is of two ways process involving both extracellular and intracellular digestion. The primary lysosomes secretes hydrolysis, very important enzyme, into the extracellular matrix by exocytosis, resulting in partial degradation of the matrix. The, the incomplete products of digestion are absorbed into the cell by endocytosis. Digestion is then complete in the secondary lysosomes. Extracellular digestion takes place during uh, during bone erosion process. The important thing is that intracellular digestion may involve autophagy and heterophagy. Autophagy means it is intracellular digestion in which enzyme produced by the cell break down molecules and pieces of cytoplasmic materials from the cell itself. Next point is that heterophagy. It is the intake of exogenous material into the cell and the subsequent breakdown of this material by enzymes by the process of endocytosis. Heterophagy are two types, namely phagocytosis and pinocytosis. Just it play very important role in the hormone secretion. There are evidence that lysosomal hydrolyses are involved in the secretory activity of some cells. The function in modifying the products synthesized by the cell before secretion, and also in the regulation of secretion. Just you can see there is a secretion of the T3 and T4 hormones. Now, it also helps the very important function in the fertilization. During fertilization process, uh, acrosome means gant lysomes of sperm head ruptures and releases enzymes on the surface of the egg. These enzymes digest the egg membrane and provide way for the entry of sperm nucleus into the egg. Another important function is that just you can see that similarly in the diagram, this is the sperm, and at the tip of there is a presence of the lysosomes, and just coming in contact to the just here and digesting the membrane, and sperm egg cell are moving towards the egg cytoplasm. It also play very important role in the autolysis. Autolysis means in case of the morphogenesis. Autolysis refers to the digestion of own cells by the lysosomes. Autolysis occurs during the amphibian metamorphosis and insects metamorphosis, etc. Lysosome just it helps in the breakdown of the chromosomes. Lysosome contain the enzymes deoxyribonuclease, DNase, that is called DNase, which attacks DNA and causes chromosomal breaks and rearrangement. Thus, release of hydrolysis from the lysosomes can in result structural abnormalities in chromosomes that can lead to cause disease like cancer, etc. Thank you very much.